looking back, looking forward, looking down, looking up, looking around. Come, join a journey of exploration to the distant past, to the frontiers of tomorrow, from the infinitely small to the infinitely large, the mysterious, the mundane. Come, let us travel to the horizons of knowledge. This program has been made possible thanks to a generous grant from the Swiss Agency for Development and Cooperation, the Vodafone Egypt Foundation, the OPEC Fund for International Development, and the Carnegie Corporation of New York. This is one of the shortest scientific papers ever written. It's only 1 page long. It appeared in the April 1953 issue of Nature. It was produced by Watson and Crick, and as you can see here, it has a signature by the author, Jim Watson. It appeared in the 1953 issue of Nature, and it won its authors a Nobel Prize. It is considered one of the most important discoveries of the 20th century. It revolutionized science. It yielded a dizzying array of new sciences, from genomics to proteomics to cell biology. It opened up the way to modern biotechnology. Yet, this little paper of 1 page with only one little diagram, the famous double helix. In that paper, Watson and Crick had correctly described the structure of DNA and effectively explained the mechanism of heredity. Humans had found the building blocks of life. So the discovery of DNA was to become a pivotal event in the history of biology. In fact, it marks the transition from classical biology to modern or new biology. But leaving aside the structure of DNA for a moment, let us go back and recall that classical biology had been built first around the taxonomy of Linnaeus, then by the theory of evolution of Darwin, and then by the enormous contribution of Gregor Mendel. Mendel prefigured much of the new biology. Not only did he establish the rules of heredity working with his pea plants, but he was the first to suggest a factor that would later become known as a gene. Mendel's concept was given a name by Hugo de Vries in 1889, who coined the term pangen, stands for the smallest particle representing one hereditary characteristic. Now, William Johansen abbreviated this term from pangene to gene, gen in Danish and German, about 2 decades later.
A gene is kind of the basic unit of our kind of inheritance. So, for example, each gene has a kind of a trait that's connected to it, whether it's our eye color or our hair color or whether we will or won't get a disease.
A gene is a piece of DNA that commonly codes for a single protein. Even that definition is increasingly complicated because we now know that genes can code not only for a protein, sometimes they code for several proteins, and sometimes they code for little molecules called not DNA, but RNA.
So now we had genes, whatever they were, recognized as the key agents of heredity. They resided in the cell's nucleus and somehow transferred traits according to certain rules. But where were they in the cell? What were they made of? How did they replicate and transfer traits from the mother cell to the daughter cell when cells split? How did they compose themselves when parents produced an offspring? In 1910, Thomas Hunt Morgan showed that genes reside on chromosomes. In 1944, Oswald Avery, Colin Monroe McLeod, and Macklin McCarthy showed that DNA holds the gene's information. Aha! DNA now appears on the scene. It stands for deoxyribonucleic acid, a strange molecule. It is a long and thin molecule wrapped around itself many times over in a very, very complex set of coding that carries within it the secrets of life and heredity.
DNA is a chemical. It is a long, long polymer. Think about it as a hair, but think about it as two spirals wound around each other.
DNA is the repository of information that every living organism contains. And it is the place where the instructions are laid down. It is material that can be copied. It's material that can be read by the cell to make all the proteins and the other chemicals that a cell needs. So DNA is essential to almost all forms of life.
By the late 1940s and the early 1950s, searching for DNA and explaining its structure had become an exciting competition among some of the brightest minds in the field. The exciting final chapters of the race to discover the structure of DNA were played out in England.
A brash young American, James Watson, joined Francis Crick in Cambridge in UK, where they started building physical models of how such a molecule could look like. But they lacked the knowledge of the structure of molecules for their models to come out right. They had several unfortunate efforts. The required knowledge was being developed nearby at King's College London, where the fascinating Rosalind Franklin worked with or competed with her colleague Maurice Wilkins. Rosalind Franklin, one of the most brilliant and gifted scientists of her generation, was into crystallography. She was basically taking X-ray pictures of molecules of DNA with light channeled through a crystal. Her photos would show that the structure of DNA was a helix, and between 1951 and 1953, Rosalind Franklin came very close to solving the mystery of the structure of DNA. Close, but not quite there. At one point, Wilkins showed Watson one of Franklin's portraits of DNA made with crystallography. When Watson saw it, the solution became apparent to him, and he rushed back to Crick in Cambridge to discuss it.
At the middle of the century, two now very famous scientists, Watson and Crick, published a little paper that said DNA has the following structure. It's a double helix, and oh, by the way, the two strands of the helix can open up and direct the synthesis of a new helix, and that's the basis of genetic heredity. That goes back to Darwin.
The great discovery by Jim Watson and Francis Crick of the structure of DNA. That DNA was a double helix. There were two strands that were held together by pairing of the four elements that compose DNA, things we call bases, and those bases come in four letters, A, C, G, and T.
Yes, Watson and Crick figured out what component of a cell actually contained the hereditary information. And that was important in a mechanistic sense, and it's hugely important technologically.
In my own work, we try to study cancer, for example, by taking either cells or experimental animals like mice and changing them by manipulating the genes in the cells or the animals. And we do that by using a kind of technology that only became available about 20 years after Watson and Crick had done their work.
Now, the new model that Watson and Crick would build would be just right. It was a double helix, and it looked like a ladder that had been twisted. The external parts, the rigid parts of the ladder, the stringers, were made of phosphates, and the inside rungs of the ladder were made of pairs of bases. Each rung was made of two bases, C with G, T with A. The bases never matched any other way. Now, this also immediately explained how the DNA molecule could replicate so accurately. It unzipped like a zipper, and because of the specificity of the bonding, A with T and C with G, each of the two sides would recreate a complete DNA molecule exactly like the original with no errors. Thus, the hereditary material could be passed on completely from the mother cell to the daughter cell during cell division. The results of this model were published by James Watson and Francis Crick in Nature almost immediately. Maurice Wilkins and Rosalind Franklin each had a supporting article. Franklin died a few years later in 1958 at the tender age of 37. 4 years later, in 1962, the Nobel Prize was given to Watson, Crick, and Wilkins. Many scientific discoveries followed. But one of the great scientific enterprises and another epic race was to take place before the end of the century: the sequencing of the human genome. The human genome, that is the DNA that is in each of our cells, the entire genomic information available in each cell. In fact, that single molecule contains the entire blueprint of how to build that person. After all, we all start from one fertilized egg, one cell. That cell then divides again, and then definition begins. Some cells start becoming bone, others become tissue, others become blood. But if their appearance and functions change with the development of the fetus, all the cells and every cell contain the entire DNA molecule as it was in the fertilized egg. Now, where does this genetic material come from? Half of the genetic material comes from the father and half from the mother. DNA is arranged into 23 pairs of chromosomes, which are really bundles of curled up DNA. And the last pair are the sex chromosomes. They are called X and Y chromosomes. the X chromosome comes from the mother in the egg. The second chromosome comes from the sperm. If it too is an X chromosome, then the baby will be a girl. If it is a Y chromosome, then the baby will be a boy. The sperms are approximately 50% each way, so the odds are reasonable that the future generation will be about evenly matched. This story shows that contrary to long-held beliefs by various husbands, especially kings who wanted male heirs for their thrones and searched for spouses that would deliver them boys, were wrong. The sex of the child is determined by the husband, not by the wife. But let's come back to the human genome. Just how big is the human genome? Well, it needs to be answered by analogy. If the DNA was stretched out, if all the 46 chromosomes were stretched out, connected end to end and straightened, we would have a length of about 2 meters and a width of about 2.4 nanometers. A nanometer is a billionth of a meter, a millionth of a millimeter, which means a human hair is almost 50,000 times thicker than a DNA molecule. So given the length of the molecule and its very narrow width, that is the equivalent of a ribbon 10 meters wide that is so long that it could wrap around the Earth 2 million times. If I were to say that the stretched out DNA material in one cell could be approximately the height of a tall man, about 2 meters, with 100 trillion cells in the human body, if the DNA in each cell is taken and added end to end from all the cells, it would stretch from here to the moon and back 8,000 times. Wow. On that molecule lies all our genetic material, the human genome. Now, the human genome is about 3 billion base pairs. If we could sequence it and have every bit of it mapped, we would be able to identify every gene on every chromosome. We would be able to match genes with what each one does, and we should be able to understand why genetically inherited diseases occur, and maybe we would be able to redesign medicine and do many wonderful things.
Many scientific discoveries followed. But one of the great scientific enterprises and another epic race was to take place before the end of the century: the sequencing of the human genome.
So a wonderful international collaborative enterprise was started. It was called the Human Genome Project. Initiated by the same James Watson, who was co-discoverer of the structure of DNA in 1953, James Watson at the NIH initiated this, and it was to be continued by others and completed under the direction of Francis Collins. It was a public research project in which many scientists from all over the world participated. The technology was slow and ponderous, but it was a truly international effort, with all scientists working for the benefit of humanity. It was expected to cost about $3 billion and take 15 years to complete. Enter another brilliant and brash figure, Craig Venter, who in collaboration with Nobel laureate Hamilton Smith developed the so-called shotgun technique to sequence genomes. That enabled him to accelerate the process enormously by relying on parallel processing by powerful computers and then reassembling the results from all these parallel computing sequences.
So Craig Venter himself is a fascinating person because he was the person that individually sequenced the human genome, and he's a very brilliant guy. Now he's working on these synthetic creations of new life forms. The purpose of it is not to create monsters. The purpose of it is to create, for example, a kind of a bacteria that might eat up bad things in our environment. So could get rid of waste, for example. Another purpose would be maybe to convert crops that are completely useless, like weeds, into new energy sources. So the good side of what Craig Venter is trying to do is quite exciting, actually.
Venter created Celera Corporation, a private sector effort to sequence the human genome with a view to do it for $300 million in 3 years. Now the race was on. The race was declared a tie when both the private group and the public group published their genome maps in February 2000 in Nature and Science. They were both welcomed by President Bill Clinton in the White House, and the cover of Time magazine shows them both together. Bitter rivals had crossed the finish line at the same time. So now we had mapped the human genome. But it was just the beginning. We needed to explain all those letters, 3 billion of them. Only small parts of the genome are identifiable gene sequences that actively code for and create the proteins that allow us to be the living organisms that we are. And surprise, surprise, we discovered that humans had only about 20,000 to 25,000 genes, not the 100,000 or more that had been expected a few years before. How could such a complex organism have so few genes? After all, yeast has about 6,000, fruit flies have about 13,500, mice have about 25,000, plants such as wheat have about 40,000 genes, worms have as much as 20,000 genes, and we, human beings, have about 20 to 25,000 genes, somewhat more than a worm, but about the same as a mouse. And what is more, we share over 98.5% of our genome with chimpanzees. The difference is a tiny 1.2%. It is another blow to the ego of humans. It is obvious that what we thought we understood as the manner in which DNA codes for proteins is not correct. Life is much more complex than the simplistic models we had. It was becoming rapidly clear that far from being the final answer to everything, mapping the human genome, or any genome for that matter, is just the beginning of the real journey of discovery and understanding. We do not yet understand all the complex interactions that make life function, or how these building blocks of life actually work, even at the level of what we consider lowly organisms, much less at the level of humans. Yet, that did not prevent us in the last 20 years from racing along in many different enterprises. An entire new industry was created, biotechnology. Some see this new technology as a savior, others are afraid of its potentially enormous impact. But it is here, with applications from cloning to genetically engineered plants, from biorobotics and cyborgs to gene therapy. Whole new generations of scientists and businessmen are vying with each other to create the future. How that exploding knowledge is affecting our lives and how humanity is coping with the new ethical challenges that arise is a truly exciting story. But that is a story for another day. I am Serageldin, and I thank you
for having joined me in this episode of Horizons.